The trillions of cells that make up your body all came from a single cell. You started as an embryo with just one cell, which then split into two, then those two into 4, 4 into 8, 8 into 16, 16 into... This splitting process is controlled by the genes of each cell. They use the molecular version of a stop-go sign to tell the cell when and when not to split. As fully grown humans, it's best if the cells stay in the stop phase, but occasionally you need your cells to switch back to go in order to replace the cells that you lose on a day-to-day basis, or when your body needs to regrow, such as when you have a cut or wound that needs healing. Occasionally, this stop-go signal starts working incorrectly, meaning a cell can start splitting rapidly in an uncontrolled way. The resulting cluster of cells creates a tumour, the kind we might see in breast cancer. Different types of cancer can arise in different ways, but here at Guy's Cancer Centre, we're investigating the role that our immune systems might play in helping breast cancers to grow. The immune system is a vast network of lots of different types of white blood cells that move all around our bodies, spotting foreign invaders. This normally involves spotting things like bacteria or viruses, but what's incredible is it can also detect cancerous cells. When white blood cells spot cancer cells, they produce chemicals that start an inflammatory response in the surrounding tissue, which in most cases help to destroy the cancer. However, in some cases, the cancer cells cover themselves with molecules that change the way white blood cells interact with them. These molecules on the cancer cells are called glycoproteins. Glycoproteins are branch-like structures made from a mix of sugar and protein. There are 3 key differences to these glycoproteins that commonly occur in cancers: length, quantity, and direction. Number 1. Length. In healthy cells, the sugars in the glycoproteins arrange themselves into long chains, but in cancer cells, the sugars are short and stubby. Number 2. Quantity. In cancer cells, the amount of glycoproteins increases massively, with as much as 400 times as many appearing on the surface of each cell. Number 3. Direction. Normally, glycoproteins line the channels and ducts within our bodies, acting as a defensive layer of your digestive tract, or the air sacs in your lungs, where the chance of infection from outside invaders is more likely. Another place they appear is the duct within the breast tissue. However, in cancers, they also point inwards towards the body, meaning they can interact with cells they never normally meet, including immune cells. When a particular type of white blood cell known as macrophages detect a large amount of short sugar glycoproteins inside the body, they incorrectly identify the cancer cells as healthy cells that need support, like those in a wound, cut, or graze. The macrophages then start providing the cancer cells with nutrients and other helpful chemicals that help the tumour to grow, while also protecting them from other white blood cells who might be trying to destroy the cancer. By analysing these 3 factors about the length, quantity and direction of glycoproteins and their effect on immune cells, our Medical Research Council funded research team here at King's College London hope to gain a better understanding of how our immune system interacts with breast cancer cells to grow and spread, to help find new ways of diagnosing and treating breast cancer.